Good day, everyone. We are here to present to you our topic in biotechnology products entitled DNA fingerprinting. But before that, we would like to introduce ourselves to you. I am Robles Princess Joycey of BSHM.
I am Hanna Maleen Matanguihan from BSHM2.
Soien Mendoza from BSHM.
Chandran Andrei Joshua from BSTM.
Marc and Rubinos from BSHM2.
DNA fingerprinting. What is DNA fingerprinting? DNA fingerprinting is a way to identify a certain individual, rather than simply identifying a species or a particular trait. A technique used by scientists to distinguish between individuals of the same species using only samples of their DNA. It allows DNA samples from different people to be compared to look for similarities and differences. It is used for solving crimes. Who invented the DNA fingerprinting? A Leicester University geneticist Alec Jeffreys developed a technique called DNA fingerprinting in 1985. Historically, identity testing in the forensic field started with the analysis of the ABO group system. ABO group systems, the classification of human blood based on the inherited properties of red blood cells as determined by the presence or absence of the antigens A and B, which are carried on the surface of the red cells. And the four main types of ABO are identified as blood type A, B, AB, and O. Your blood group is determined by the genes you inherit from your parents. Professor Jeffreys was interested in studying the genetic variation between individuals and had done some of the early work to detect genetic differences in humans. However, the answer did not come to him on the initial project he was interested in, but rather on an unrelated project. Analysis of the myoglobin gene in seal meat at the headquarters of the British Antarctic Survey. When he and his colleagues compared the myoglobin gene in seals with the human counterpart, they found that some short repeating sequences were homologous between seals and humans. When they compared those sequences with the published sequences of tandem repeats called mini-satellites, they found that they were the same. Professor Jeffreys recognized that the repeating sequences could be highly variable informative genetic markers. His group developed a radioactive probe made up of short sequences that could latch onto those repeating sequences and ultimately reveal patterns that were unique to each individual, a DNA fingerprint. Biological samples used for DNA fingerprinting: blood, hair, saliva, semen, body tissue cells. DNA samples have been obtained from vaginal cells transferred to the outside of a condom during sexual intercourse. There are 8 steps for DNA fingerprinting: isolation of DNA, digestion of DNA by restriction endonucleases, electrophoretic separation of different fragments, transfer DNA on nylon nitrocellulose membrane, probing or probe labeling, hybridization, autoradiography, interpretation of band patterns.
Isolation of DNA. DNA must be recovered from cells or tissues. Only a small amount of blood, hair, or skin needed to isolate DNA. Digestion of DNA by restriction endonucleases. The DNA is cut into fragments using discretion fragments. Each restriction enzyme cut DNA at a specific base sequence. The Section of DNA that are cut are called restriction fragments. This yields 1,000 restriction fragments of all different sizes because the basic sequence cut may be far apart. Commonly used by RE, reaction mixture is incubated overnight at 37 Celsius.
Electrophoretic separation of different fragments. Fragments are separated on the basis of size using a process called gel electrophoresis. DNA fragments are injected into wells and an electric current is applied along the gel. DNA is negatively charged, so it is attracted to the positive end of the gel. The shorter DNA fragments move faster than the longer fragments. DNA is separated on basis of size. Transfer DNA on nylon nitrocellulose membrane. The DNA fragments are transferred to a nylon sheet by placing the sheet on the gel and soaking them overnight by the process Southern blot.
Probing or probe labeling. It is adding radioactive or colored probes to the nylon sheet, which is complementary to target sequences. Each probe only sticks to one or two specific places on the sheet. Next is hybridization. Hybridization. Labeled probe DNA should be hybridized with the complementary sequences located on nylon membrane for the detection of position of later. Membrane is washed to remove non-specific binding and clearing of the background.
Autoradiography is used to detect the sequence of genome bound with a hybridized radioactive probe on membrane. The technique involves alignment of hybridized membrane with X-ray film. The X-ray film after its development shows multiple number of bands that look like barcodes and known as DNA fingerprints. Interpretation Interpretation of band patterns, analysis of band patterns of different individuals, comparison of position of bands. Computer software are also available for the analysis of DNA fingerprints.
Uses of DNA fingerprinting. Diagnosis of inherited disorder and crime. Diagnosis of inherited disorder helps diagnose disorders in both prenatal and newborn babies. Disorders may include cystic fibrosis, hemophilia, Huntington's disease, familial Alzheimer's, sickle cell anemia, thalassemia, and much more. In crime, forensic science is the use of scientific knowledge in legal situations. The DNA profile of each individual is highly specific. The chances of two people having exactly the same DNA profile is 30,000 million to 1, except for identical twins.
Genetic profiling as a forensic tool. At this time, the forensic implication of genetic fingerprinting were emerging. The original process proved to be inadequate for this. And so, from 1985, Sir Alec and his team developed a variation which they call genetic profiling for forensic use. Again, its first application caught the public mood. Two young girls were raped and murdered in the Enderby area of Leicestershire. A man who had been arrested had confessed to one murder, but not the other, and the police decided to use genetic profiling thinking to prove him guilty of both cases. Against all expectations, he was found to be innocent of both. Then the hunt was on to find a genetic profile among the entire male population of the area that matched sample taken from the two victims. No match was found until Colin Pitchford was overheard boasting of he was he had persuaded a friend to give a sample on his behalf. The case was solved. DNA fingerprinting, one of the greatest discoveries of late 20th century, has revolutionized forensic investigation. This review briefly recapitulated 30 years of progress in forensic DNA analysis, which helped to convict criminals, exonerate the wrongly accused, and identify victims of crime, disaster, and war. Current standard method based on short tandem repeats (STRS) as well as lineage markers Y chromosomes, mitochondrial DNA are covered and application are illustrated by casework example. Benefits and risks of expanding forensic DNA database are discussed and we ask what the future holds for forensic DNA. But before that, let's look back to the history of DNA fingerprinting. The history of genetic fingerprinting. You could say that the path that led to the discovery of genetic fingerprinting began for Professor Sir Alec Jeffreys when his father gave him a chemistry set, a microscope at the age of 8. Scientists, he believes, like musicians, not bo- are not born but made. Nonetheless, he acknowledged that this parental encouragement played its part in his choice of vocation. It might have been disastrous, as he explained. This was a real chemistry set, not a child's toy. And later, when I was 13 or 14, I got a face full of sulfuric acid, which is why I wear this beard now. I consider it a badge of honor. That's Sir Alex's words. The Eureka moment. This is Sir Alex's word. My life changed on Monday morning at 9:05 a.m. September 10, 1984. What emerged was the world's first genetic fingerprint. In the science, it is unusual to have such a Eureka moment. We were gathering extraordinarily variable patterns of DNA, including from our technician and her mother and father, as well as from non-human sample. My first reaction to the result was, this is too complicated. And then the penny dropped and I realized we had a genetic fingerprint. It opened up a new area of science. The research team immediately grasped its application, including crime, maternity, and identical twins, as well as work on conservation and diversity among non-human species. Later that day, Sir Alec wife added another to the list, immigration. That was when I realized this had a political dimension and that it could be changed the face of immigration dispute, especially where no documentary evidence existed.
DNA profiling. DNA profiling is a sensitive technique which only need a few skin cells, a hair root, or a tiny amount of blood or saliva. There are sections of loci of chromosomes where instead of a gene consisting of a long sequence of bases, there are much shorter sequences of 3, 4 or 5 bases that are repeated many times. For example, samples are repeated are called short tandem repeats or STR. Short tandem repeats or STR are short repeated sequences of DNA that account of approximately 3% of the human gene. The number of repeat units is a highly variable among individuals, which offers a high power of discrimination when analyzed for identification purposes. At these places, the chromosomes where we find these STRs are areas that vary in number of repeats. DNA profiling looks only at these STRs. As a sample is collected, this could be from some blood at a crime scene or swab from the inside of someone's cheek. For example, the DNA is then extracted from the sample. Many copies of this DNA may be made using the polymerase chain reaction or PCR. Special enzymes called restriction endonucleases are used to cut the DNA up into different size pieces. Polymerase chain reaction or PCR is a laboratory technique used to make multiple copies of segment of DNA. PCR is a very precise and can be used to amplify or copy a specific DNA target from a mixture of DNA molecules. The mixture is then cooled so the primers annealed or bind to the DNA template. The DNA samples are then put into wells in a special gel called agarose from the process of gel electrophoresis, which separates the DNA fragments by size. The pattern is then transferred to a nylon sheet in Southern blotting. And finally, the lines produced by DNA samples from different people are compared.
A DNA molecule consists of a big long chain of smaller molecules called nucleotides. Each nucleotide contains a section that can be one of four different even smaller subunits. Each of these is denoted by the letters A, T, C, and G. This means that DNA is a big long string of these letters in specific sequences. The order of these letters is a set of instructions telling our cells how to build proteins, which is the key to life.
Advantages and disadvantages of DNA fingerprinting. 1. DNA fingerprinting provides another layer of forensic evidence. A pair of gloves might be able to stop fingerprints from being left behind at a crime scene. DNA evidence is much more difficult to prevent. 2. It offers a greater level of certainty than standard fingerprinting. No two people are believed to have the exact same fingerprints, just like no two people are believed to have the exact same DNA. To compare fingerprints for matching, specific points of comparison are logged, either by a visual examination or analytic software, to determine its accuracy. 3. DNA fingerprinting is unobtrusive. Unobtrusive means not conspicuous or attracting attention. Traditional fingerprinting requires dark ink to be placed on each finger. That finger is then rolled onto a card. Some fingerprinting may require palm prints as well. In comparison, DNA fingerprinting requires a simple cheek swab to gather the information that is required. 4. The evidence collected from DNA fingerprinting can be stored indefinitely. DNA fingerprinting creates a specific genetic profile that can identify an individual. These profiles can be stored in databases for an infinite period. Because this information is transformed into data points, it can be communicated quickly over the internet. 5. DNA fingerprinting does not require a specific sample size. The genetic materials that are obtained from a DNA sample can be amplified with current technologies. That means a sample a small sample provides the same comparison data as a large sample provides. These are the disadvantages of DNA fingerprinting. 1. DNA fingerprinting is still an imperfect science. Having a technology that is 99.9% accurate may seem like a reliable technology. When that percentage is applied to real-life cases, however, it indicates that there may be one error in every 1,000 cases that are analyzed. 2. It requires information obtained to be properly interpreted. Let's say that a major crime is committed at your local art museum. A famous painting worth 300 million has been stolen. You just happened to be at the museum the day before, looking at the exact painting. Maybe you put your hand on the wall next to it, or maybe you had an allergy attack and sneezed a few times into a tissue, which you then threw away. Your DNA is going to be collected there, and you are going to be a suspect. 3. DNA fingerprinting information could be stored without personal permission. When infants are tested for PKU in the United States, a blood sample is placed onto a filter paper card. The dried blood on these cards can be useful to maintain high levels of quality control at a laboratory. They can also provide public health benefits and offer assurance monitoring. 4. DNA fingerprinting relies on human accuracy. DNA fingerprinting may provide another layer of evidence for enforcement officials when building a case. It also relies on human judgment to create results that are available for interpretation. If an error occurs during the processing technique, then the results from DNA fingerprinting may not be accurate.
Technique for DNA fingerprinting. AFLP technique. AFLP technique is amplified fragment length polymorphism. It is a new technique on how to get a DNA. This is a technique based from PCR, which is the polymerase chain reaction which tests to detect a specific organism inside our body. It has a three-step to do for us to achieve the target fragments to distinguish our DNA. But there are restrictions in doing amplification from trunking nucleosides to primary extension can cause a high number of restriction fragments. The AFLP is the most powerful in detecting our fingerprinting, which already used in other countries. Restriction digestion, adapter ligation, preamplification, selective amplification, gel electrophoresis. Restriction digestion, the process of cutting DNA molecules into smaller pieces with a special enzyme called restriction endoclysis. Adapter ligation is a method of attacking synthetic or oligonucleotide with known sequence to both or either ends of complementary DNA fragments. Amplification is a method used to increase the concentration of specific panel of targets in a simple prior up to PCR analysis, reducing the required sample input for multi-target QCPR experiments. That's all. Thank you.